These are my disclosures with potential conflicts from both governmental and industry sources. I work a fair amount with industry doing clinical trials. So just to set the context, as of a couple of days ago, there were over 140 million cases and over 3 million deaths due to this pandemic, which does not seem to be slowing down at all. Vaccines development began almost immediately after the pandemic was declared. The World Health Organization put together a target product profile to say what anybody who is developing a vaccine should be targeting, which is why when one looks at the various vaccines and their efficacy, the target efficacies, they were all the same. And this because all manufacturers were given guidance of what the preferred vaccine would look like and what the minimal requirements would be in order to get a vaccine authorized for use. Typically, a vaccine takes between 10 and 15 years to get approved, where one first selects the target, one then has preclinical studies, clinical development, and then the approval process. But this is a timeline that just is not would not have been viable to meet and address the pandemic. But all the steps of vaccine development had to still be followed in order to develop the vaccine. And that starts with vaccine discovery, which is basic science, where one understands what the virus is and determines how it infects and does its damage to humans and how the immune system responds. Then one picks a target antigen, decides how one's going to present it to the immune system, and decides whether an adjuvant is going to be required. Now, fortunately, a lot of the vaccine discovery work had already been done because of other coronaviruses, SARS and MERS. So there was a lot of work done that had already identified the spike protein as an important target, and vaccines had already started to be developed for those pathogens. So the vaccine discovery part was efficiently moved along. But once one has the target, one then moves into preclinical studies. And these are studies in animal models where one looks at the effect of the vaccine on various vital functions, cardiovascular, respiratory, central nervous systems, looks at toxicity, looks at the effect on different mammalian species. If it's going to be used in pregnancy, one needs to look at genotoxicity and reproductive toxicity. One needs to look at carcinogenicity. If it's going to be used in children, one needs to do pediatric studies as well. And if there's an animal model, one wants to look at efficacy. Now, again, some of this work had already been done. So animal models had already been determined for other coronaviruses. So laboratories were able to get up and running very quickly. Some of the things that were done that we're paying some of the price for now is that in order to expedite the process, some of the reproductive toxicity studies and juvenile studies were not done, which has now delayed the ability to use vaccines in children because some of this work still has to be done and had to be done. And we're just now getting into clinical trials that involve children. Once one has a vaccine candidate that's passed all of its preclinical work, one then starts the vaccine clinical trial process. And the first trials are phase 1 studies, where one's primary outcome is safety and getting an early look at the immune response. Phase 1 studies are typically small, and they're in the tens, so anywhere from 40 to 80 or 90 participants in a phase 1 study. And these may be controlled or uncontrolled. If they're controlled, oftentimes a placebo. Once one has some preliminary data on the safety, one then goes to phase 2 trials, which looks more at the immune response as the primary outcome while gathering more data on safety. Outcome measures here might be antibody titers, injection site and systemic adverse reactions, and cellular immunity. Phase 2 studies typically are in the hundreds, so anywhere from 200 to 300 up to 800 or 900 participants. And these are typically randomized and blinded. If there's a licensed product, one has a comparative study. If there's none, then one has placebo-controlled. After phase 2 studies, one does phase 3 studies, and these studies are to determine whether the vaccine works. Now, while one's doing this, one gets more information on immunogenicity, one gets more information on safety, but the primary outcome is the disease. The target population for phase 3 studies are the population in which the vaccine is going to be used. The size of it really depends on the frequency of the disease. And because of the target profile, most vaccine studies that you've seen needed 150 outcomes. And for 150 outcomes, most of the sample sizes were 30 to 40,000 people. So large studies, and these studies had to be done in areas where one was seeing a lot of cases. Which is why there are no phase 3 studies that have been done in Canada. Phase 3 studies are randomized and blinded, and typically in a case where there's no vaccine, placebo-controlled. Back in April of last year, the New York Times ran a nice article calling "How long will a vaccine really take?" And this looked at the 10 to 15 years, saying that a vaccine would probably be available sometime in 2035, 2037. Not really helpful. And then they looked at saying, how could timelines be compressed, including how could the clinical trials be compressed? And this is where one got that estimate back a year ago that a vaccine could potentially be available as early as 12 to 18 months. Now, amazingly, we've been able to exceed those expectations, and vaccines are actually available in 8 to 10 months. So how does one do that? First, from the clinical trial standpoint, one gets into adaptive designs. An adaptive design is a clinical trial that designs for prospectively planned changes and modifications to one or more aspects of the design based on accumulating data from participants already in the trial. And adaptive designs are useful because they increase the efficiency and speed, they reduce the cost, and because one can eliminate non-performing arms quickly, they're of benefit to the participants as well. On the top here, the design of a typical clinical trial is you design it, you conduct it, and then you analyze it, and then you go on and design another trial. In an adaptive trial, you design it, you conduct it, you review, you adapt one or more of the arms, and you can have multiple of these cycles before you do the final analysis. And that saves a substantial amount of time. In Canada, and in most places, phase 1/2 adaptive trials have been highly preferred. And this increases the efficiency without sacrificing safety. And within a single trial, then one can explore multiple different antigens, different adjuvants, different formulations, different target populations. So starting with healthy young adults first, and then moving into older adults, and then into the elderly, without stopping and starting a new trial. And based on the interim analysis of previous steps, one can go on to the next steps. And this is what an adaptive phase 1/2 trial looks like, where you might have in the blue bars what would typically be a phase 1 study, in the red bars a phase 2 study, in the green bars a phase 3 study. And if one put these all end to end, it would be about 3 years of time. Whereas here you can go from one formulation to the next. And then as soon as you have information on each of those age cohorts, one can then start the phase 2 trial while you're still doing all the follow-up of the phase 1 trial. And likewise, start the phase 3 trial even weeks to months after you start the phase 2 trial, and then do follow-up simultaneously. And this is how all the manufacturers got into phase 3 as quickly as they did. This is a nice graphic that you can download. It's updated from the WHO that looks at the status of the various vaccines and platforms that are underway. And I don't expect you to read this off this. I've summarized it in this slide. And as of April 20th, so a couple of days ago, There were 275 or so candidates in development. 91 of them are in clinical trials. 4 are what we say is phase 4, so they're being widely used. 15 are now in phase 3, and 6 are in phase 2 and 3. And of these, 11 vaccines are being developed in Canada. Most of the vaccines can be summarized or categorized into 6 major platforms, which I'm going to go into detail on the next several slides, so that one can understand that while there are 275 vaccines, many of them are similar. Virtually all of them are targeting the spike protein. When Canada chose which vaccines it was going to purchase, it made sure it had vaccines amongst each of these most of these platforms. So the first type of vaccine one can have is a live attenuated vaccine, where by serial passage or direct specific mutations, one can then weaken the virus. And these are the type of vaccines that we use for MMR, varicella, polio, the nasal flu vaccine, rotavirus, yellow fever. And there are 1 or 2 companies that are looking at attenuated SARS-CoV-2 vaccines. They have not reached advanced stage at this time. Killed inactivated vaccines are the oldest type of vaccine we have, where the organism is grown in culture and then is killed. And it's killed using one of a number of different mechanisms. That can be chemical or heat, or a combination of them. And the organism can no longer replicate, but their proteins are left sufficiently intact to be recognized by the immune system and evoke an immune response. So the vaccine, because it's been inactivated, is not infectious. The old wholesale pertussis vaccine that we used to use in Canada is this type of vaccine. The inactivated flu vaccine. We have a cholera vaccine like this, the inactivated poliovirus vaccine. And there are inactivated vaccines. A number of the Chinese vaccines that have been developed are this type of product. We can also, a little bit more refined, our purified subunit or inactivated subunit vaccines. And this is where one produces the organism, grows the organism, and then you purify what you've identified as protective antigens from the pathogen. These can be proteins or polysaccharides. Because you don't have as many of the other products of the organism there from a killed vaccine, you have a lower risk of adverse events, of side effects, but it may be less immunogenic, because as you purify a protein, you lose its tertiary structure. And as you get smaller and smaller subunits, such as peptides, it may be less immunogenic. And therefore, you may need to use an adjuvant. Acellular pertussis vaccines, the Haemophilus influenza conjugate vaccine, meningococcal and pneumococcal vaccines are all this type, as are the tetanus, diphtheria, and pertussis toxoid vaccines. Now, these can be refined rather than growing the organism and purifying the antigen. You can also use recombinant purified subunit vaccines, and there are a number of these that are being developed for SARS-CoV-2. Hepatitis B vaccine is a good example of one that we use routinely. That's that uses this platform. Virus-like particles is a relatively new, but now getting old technology, where the viral proteins are derived from the structural proteins of a virus, and where the proteins can then self-assemble into particles that resemble the virus, but they lack any of the viral nucleic acids. So there's no genetic material, so they can't reproduce. They are not infectious. But because of their very highly repetitive multivalent structure, and these virus-like particles look just like a virus, so they're typically more immunogenic than the subunit vaccines. And HPV is this type of vaccine. And there are a number of SARS-CoV-2 vaccines being developed. As one Canadian technology, the Medicago vaccine, which is just entering phase 3 studies now, is this type of vaccine platform. Vectored vaccines are another ones that you've heard a lot about, where a non-virulent or attenuated virus is used to express the antigens of a heterologous organism to induce immunity. There are a number of viruses that have been used for this. Adenoviruses are ones that you're hearing the most about, but vesicular stomatitis virus was used for the Canadian Ebola virus vaccine, for example. Yellow fever vaccine strain has been used. Even the measles vaccine strain has been used. And you can also do this in bacteria. So there are a number of companies that are starting studies looking at Salmonella typhi vaccine, the oral attenuated bacterial Salmonella typhi vaccine, to express SARS-CoV-2 spike antigen and have an oral bacterial vectored vaccine. The newest technology that we've heard the most about are DNA and RNA vaccines. And this is where DNA or messenger RNA deliver the nucleotide sequence that codes for the proteins that the virus uses to cause the disease, and most specifically, usually the spike protein in SARS-CoV-2. These proteins are then expressed by the host cell as antigens that the immune system recognizes and enables the body to mimic a native infection and elicit an immune response. But because it's the rest of the viral antigens aren't there, the virus can't replicate. The advantage of these DNA and RNA vaccines are that one takes the sequence and one then lets the body do all the work, the cells do all the work, and you can make very large amounts of these sequences at low cost. And one can then tailor to the immune response that you desire. Also, for variants, one can change the sequence very quickly and produce a new vaccine. There are advantages and disadvantages in comparison to the DNA and mRNA vaccines. The DNA vaccines have to penetrate both the cell membrane and the nuclear membrane. And then in the nucleus, they're transcribed into mRNA, which is then transported back to the ribosome, where the ribosome then makes the protein. Oftentimes, DNA vaccines need to have a specialized delivery system, such as electroporation. However, there are new technologies now where using viral fusogenic proteins, one of the Canadian companies is delivering DNA to the cell without a specialized technique or mechanism. It's all encoded into the same DNA. The DNA vaccines have the very theoretical risk of incorporating the DNA into the host DNA rather than just staying on the plasmid that's introduced into the nucleus. This has not ever occurred, but it's still a theoretical risk that has to be monitored for with these new technologies. mRNA does not need to enter the nucleus, so it's directly translated at the ribosome and therefore doesn't need any special delivery device. Much smaller doses are required, micrograms rather than milligrams of DNA. And then it doesn't have that theoretical risk of incorporating into the DNA, the host DNA. There are two different types of mRNAs. There are self-amplifying messenger RNAs as well as non-replicating RNAs. And both of these technologies are being developed and both are being used by some Canadian companies. Just comparing the mRNA and DNA vaccines, again, you have the delivery system may be required for the DNA vaccines, but with some of the new technology, that disadvantage is really being eliminated, particularly if these new technologies work. So where are we now with authorized global vaccines? So there are a number of vaccines that are been authorized for emergency use authorizations somewhere in the world. The four in red, Pfizer, Moderna, AstraZeneca, and Janssen have all been authorized in Canada. The Novavax vaccine has reported its phase 3 and will soon be authorized for use, we suspect, providing Canada with a fifth option. The Janssen vaccine has been authorized in Canada. Deliveries are expected within the next week or so once its evaluation for adverse events is completed by the FDA. With the phase 3 efficacy studies in general, we've been looking at what we would call reports of vaccine efficacy by press release. We had first the Pfizer vaccine, which was described as 95% effective based on 170 cases in their phase 3 study. Moderna also virtually the same, followed by AstraZeneca, which was a bit more complicated, but overall 62% efficacy, and Janssen & Janssen about 67% efficacy. Since then, we've heard additional reports of phase 3 reports. Novavax reporting 89% efficacy. Sinovac and Sinopharm, both inactivated vaccines, had efficacy that varied by location, but in the same range. There is an Indian vaccine of 80% from Bharat Biotech. CanSino vaccine, which is an adenovirus vaccine, had 65% efficacy in theirs. And the Gamaleya, the Russian adenovirus vaccine, which used an Ad5, Ad26, reported 92% efficacy. Now, since those initial press reports, we have started seeing some reports now in the last couple of months, actual full reports. So the efficacy of the Pfizer vaccine was provided in detail in the New England Journal of Medicine, showing not only the efficacy overall, but by groups, and remarkably showing that at all age levels, the vaccine still provided very high levels of efficacy. So very important information. The Moderna vaccine provided the same type of efficacy, as well as the AstraZeneca lower efficacy at around 62% overall. But if you increase the interval between the first and second dose, that efficacy did rise. And importantly, all of these vaccines were virtually 100% efficacious against severe disease and death. I just want to highlight one adverse event that is in the news now of the AstraZeneca and Janssen vaccines. It's important to put these into context, which if one looks at the background rate of cerebral venous sinus thrombosis, it's about 0.4 per million over any 2-week period, or 5 to 16 per million people per year. A study that was done, that's a pre-publication, so we still need to wait for the peer review, that looked at all the data in the US using administrative databases, suggested in the 2-week period after Pfizer and Moderna that the level of the cerebral venous thrombosis was about 4 per million people, whereas in the EMEA, the European regulator, looked at the AstraZeneca, and they looked at a rate of about 5 per million. So looking at that, perhaps after any of these vaccines, one might have that complication, potential complication. Importantly, the rate is about 40 per million people after COVID-19. So if one is going to worry about that adverse event, one needs to worry about the disease even more. For additional context, the rate of unexposed women, again, is about 4 per million per year. In after birth control pills, it's about 27 per million. So birth control pills increases the risk of the same complication, and we've been living with that for years. The mechanism is perhaps a vaccine-induced immune thrombocytopenia. The mechanism is still unconfirmed, but it's associated with low platelets and increased concentrations of PF4 antibodies. The Canadian technology, I'll just mention briefly. We have Medicago, which is in plant-derived VLP in phase 3 studies. Vito, Entos, Simvivo are all in, and Providence are all in phase 1 studies using, the Vito uses an adjuvanted protein, Entos uses DNA with a fusogenic protein, Providence uses mRNA, Simvivo uses an oral DNA vaccine. And then later we have that are still in preclinical are Precision Nanosystems, which is an mRNA, IMV, which is a peptide adjuvanted vaccine, and others are further back in preclinical from a number of different universities. There isn't, for this meeting, everybody would be interested in, what do we know about COVID vaccines and HIV? There's not a lot of information yet. The early phase studies excluded persons living with HIV. Some of the phase 3 studies included persons with living with HIV. Pfizer and BioNTech had 196 participants, but it was not analyzed. Those were not analyzed specifically in the publication. Moderna had 176. They had 0 in the COVID group, vaccine group, and 1 in the placebo. Obviously, too small numbers to look at efficacy. AstraZeneca had 160. Janssen enrolled over 1,200 and found there were 2 cases in the vaccine group and 4 in the placebo, too small again for a subgroup analysis. Novavax, we don't know yet how many were. Other companies are beginning specific studies, such as CanSino is doing a study in Argentina, which should start in the next month, who are enrolling just under 1,000 people living with HIV, and will be studying the adenovirus 5 vaccine in that population. So I'd like to just finish just mentioning about programs and implementation. As you know, NACI made its guideline recommendations on how to use the vaccines. The vaccine program started to roll out in December, and now we're in using Pfizer, Moderna, AstraZeneca. The next group of vaccines with Janssen soon to be started, and hopefully Novavax. And right now, with focusing on the first dose, most provinces are saying everybody should have a first dose by June. Right now, if one looks across the country, in the north, we have first doses been received by anywhere from 35 to 59%. Across the provinces, we're talking mainly in the range of about 20% to 25% of people have received their first doses. And these numbers are changing rapidly. Overall in Canada, we're at about 23% of vaccine coverage, which puts us gaining ground internationally, but still not at the forefront. I'm not going to talk anything about safety because Karina is going to focus on this in terms of post-marketing surveillance. Effectiveness, there are a number of studies being funded by the Vaccine Surveillance Reference Group as part of funded by the CITF, the Immunity Task Force, looking at implementation effectiveness using hospital-based surveillance systems such as the Canadian Immunization Research Network's Serious Outcome Surveillance Network, or using administrative databases such as Provincial Collaborative Network. And VSRG and CITF have also funded multiple studies in specialized populations, including hematologic malignancies, bone marrow transplant, immune disorders, and special populations like persons living with HIV. Vaccine coverage is a concern. Right now, the numbers are going up rapidly, but at some point we'll hit that mark where people become more hesitant or are more hesitant. The early adopters are obviously taking the vaccines, but we likely will need, even without variants, 65 to 70% to achieve herd immunity. So we'll have to deal with vaccine hesitancy. We know that vaccine hesitancy occurs across the world. It varies, and Canada is not immune to this at all. If one looked at a global survey of vaccine hesitancy, Canada was in the middle of the pack with about 2/3 of people saying that they would accept a vaccine. And these numbers change and need to be updated. And that's a very high priority by the public health agencies to understanding vaccine hesitancy and vaccine confidence, particularly in various populations and communities. So in summary, the development of COVID vaccines has been an extraordinary endeavor, accomplished in record time and very much achieved through global coordination. But we're only really partway through the process. Successful implementation will also require this type of focus. And while a successful outcome is increasingly likely, it's not assured. So we've been focusing on the basic science, moving to the clinical science. Now we need to focus on implementation science and social science as we get these vaccines out to everyone. Thank you.